Dr. Sage here, continuing our discussion of microbial nutrition and growth. In this video, we're going to discuss the study of microbial growth. By the end of this video, you should be able to summarize the steps of binary fission and name another method of reproduction used by other bacterial species. You should be able to define doubling time and describe how it leads to exponential growth, compare and contrast the 4 phases of growth in a bacterial growth curve, and identify one culture-based and one non-culture-based method for analyzing bacterial growth. The basis of population growth among bacteria is binary fission. During binary fission, the parent cell enlarges and then it replicates its DNA. It copies its chromosome. The cell envelope pulls together in the center of the cell, forming a septum, and then the cell separates into 2 separate cells. So bacteria use binary fission, which is their cell division, as reproduction. It goes from one individual cell, which is one bacteria, to 2 bacteria cells. Now, when we're measuring the rate of population growth, we have generation time or doubling time. This is the time required for a complete fission cycle. Each new fission cycle doubles the population. As long as the environment remains favorable, the doubling effect can continue at a constant rate. The length of generation time is a measure of the growth rate of an organism. So if we start with 1 bacteria cell, after 1 generation, we have 2 bacteria cells. After 2 generations, we have 4. And after 3 generations, we'd have 8 bacteria cells. So it doubles with every generation. Now, when we're measuring growth in a bacterial culture, we have these phases that they go through. When it first starts, it undergoes the lag phase, where there's no increase in the number of living bacteria cells. Then it enters the log phase, which is an exponential increase in the number of living bacteria cells. After this, we have the stationary phase, which is a plateau in the number of living bacteria cells. The rate of cell division and death is roughly equal. And then finally, we have the death or decline phase, which is an exponential decrease in the number of living bacteria cells. To break it down, lag phase is the flat period on the graph where the population appears to not be growing. Newly inoculated cells require a period of adjustment, enlargement, and synthesis. Cells are not yet multiplying at their maximum rate. Population of cells is so sparse that the sampling misses them. Then we enter the exponential growth or log phase. This is a period during which the growth curve increases dramatically. This phase will continue as long as cells have adequate nutrients and the environment is favorable. After which we have the stationary growth phase. This is where the population enters a survival mode, so cells stop growing or grow very slowly. Finally, we can have the death phase. Under this phase, limiting factors intensify and cells begin to die at an exponential rate, and the curve dips downward. Antimicrobial agents rapidly accelerate the death phase. Microbes in the exponential phase are more vulnerable to these agents than those in the stationary phase. Actively growing cells are more vulnerable to conditions that disrupt cell metabolism and binary fission. Growth patterns can correspond with the stages of infection. Bacterial shedding during the early and middle stages of infection is more likely to spread it to others. When we're growing bacteria, we can use a chemostat, which is a continuous culture system that admits a steady stream of nutrients. It also siphons off used media and old bacteria cells to stabilize growth rate and cell number. So it maintains the culture in a biochemically active state and prevents it from entering the death phase. How do we measure or plot population growth? Well, we have exponential growth, which is the growth pattern of microbes. It's useful to express populations of microbes as exponents or logarithms. Plotting data from a growing bacterium population. This is where we plot the number of cells as a function of time, and it can be expressed either logarithmically or arithmetically. The growth curve is a predictable pattern of growth in a population of bacteria. So how do we know the population growth of bacteria? Well, we count them. The viable count technique is traditionally used to observe the population growth pattern. The total number of cells is counted over a given time period, and it's a fundamental method of laboratory microbiology. Okay, so how do we perform a viable cell count in a laboratory setting? Well, first we take our culture we've been growing our bacteria in. We take a small amount of it, for example 1 milliliter, and add it to 9 milliliters of fresh culture. By doing this, that performs a 1 to 10 dilution. Then you take 1 milliliter of this culture and add it again to 9 milliliters of fresh culture. That will then dilute it down 1 to 100. You do it again, and you continue doing this until it's diluted down enough that you can actually visualize and see and count individual colonies. How do we visualize and count the colonies? Well, there's 2 different methods. One is a pour plate method. That's where a bacterial sample, after it's been serially diluted, is mixed with warm agar. The sample is then poured onto a sterile plate. The sample is swirled to mix and then allowed to solidify. And the plate is incubated until bacterial colonies grow. The other method is the spread plate method. That's where you take a small volume of your sample that's been diluted and you pour it onto a plate that already has solid media. You then spread the sample evenly over the surface of the plate, and then the plate is incubated until bacterial colonies grow on the surface of the medium, and then you can count those colonies. What are other methods of measuring bacterial growth? Well, another method is measuring the turbidity of the media that the bacteria are growing in. So you take something like a spectrophotometer, which passes certain light waves through your sample, and then it detects, well, how many of those light waves were able to pass through the sample. If you have media where no bacteria are growing, more of the light waves can pass through. If you have media where bacteria has been growing, the bacteria, some of them deflect or refract those light waves so they can't pass through. So a tube of clear nutrient media solution becomes cloudy or turbid as microbes grow in it. The greater the turbidity, the larger the population size. We can also use direct microscopic count of bacteria. So cells are sampled and counted microscopically. This uses a cytometer calibrated to accept a tiny sample spread over a pre-measured grid. Or we can use a culture counter and flow cytometer. Culture counter electronically scans a culture as it passes through a tiny pipette. Each cell is detected and registered on an electronic sensor. A flow cytometer is similar to a culture counter. It can measure cell size and differentiate between live and dead cells. Newer methods of analyzing population growth include polymerase chain reaction or PCR. It quantifies bacteria and other microorganisms in an environment and tissue samples without isolating and culturing them. Or you can use tests that measure ATP. This is used in food and pharmaceutical industries and may be used for rapid quantification of microbes and other environmental samples. Well, this concludes our discussion of how we study microbial growth. Until next time, this has been Dr. Sage.